was supposed to be chairing this session. Unfortunately, he couldn't make it at the very last time, so we had to change the program very fast. And, well, I guess that I was the first speaker, so I jumped to the chair. So let me remind you that we have 20 minutes for presenting our data, and then we will have like an additional 5 minutes of questions. So, well, in this case, I'm the first speaker, so well, I'm Simo Schwartz, by the way. Yeah. Okay. Okay, so thank you very much for inviting me here. Well, what I'm going to show you today is the last data that we have in one of the projects that we are still ongoing in the laboratory. It has a lot to do with the idea of sensitizing tumor cells to treatment. As you know, one of the main problems that we have in the clinical practice is that tumors eventually, many of them just become resistant to the treatment, and because of this, the rates of recurrence are quite high, depending on the tumor type, of course, but this happens. One of the reasons why tumors become resistant, and let me just say this as an introduction, is because of the presence of cancer stem cells. I've been here other times in Clinam talking about cancer stem cells, so I'm not sure that you all were there, so I will give the speech. Well, as you know, cancer stem cells are just a very small proportion, a small fraction of cells within the cancer cell, within a tumor cell, a solid tumor. And they are the ones responsible for spreading the disease. So they have an ability, which is that they are capable of surviving in non-attachment conditions. And this is one of the reasons why these cells can jump to the bloodstream and can go somewhere else, can actually invade another tissue and make generate a metastasis. So, and these cells are quite resistant to treatment in general terms. So even if you treat the tumor and you have the tumor shrinking, at some point it might be perfectly well that these cancer stem cells survive and they stay there, so eventually they grow up and you have recurrence. No? So this happens. No? So the idea of this work was to see whether we could increase the sensitivity of the cells of treatment. And this implies mainly two different strategies. One is to use more specific drugs that in somehow they hit pathways that are very relevant for cancer stem cells. So in a way that these new drugs, or these drugs, because some of them are already under clinical use, might be more useful in terms of killing cancer stem cells. The other one is how to target these cancer stem cells with nanoconjugates, which are actively targeted against some of the receptors that they have. No? So in this case, I'm going to talk more not about active targeting, because we already did, but about the use of some drugs or siRNA strategies that block and knock down a very specific pathway that seems to be involved in cancer stem cells. So let me start by talking to you about well, there's a missing slide. It should be there somewhere, but it's hiding. Anyway, it doesn't matter. One of these pathways is the pathway which is controlled by an enzyme called ALOX5. It's this one here. Okay, this one here, 5-ALOX5. Okay, this is an enzyme which is related to the arachidonic acid pathway, so it is related to the leukotriene synthesis, and it has been linked in a very indirect way to proliferation and survival in cancer. Okay, seems that somehow this pathway might be also related, or this is what's being hypothesized at this time with ALOX5, might be related to some other pathways that are very much known to be involved in cancer progression, like the Wnt pathway, for instance, and seems that it might be also involved with the way that a cell processes microRNA. So it's involved also with DICER, for instance, and so on. No? So besides that, it's not so clear how this pathway might be important for cancer stem cells and for tumor growth. The fact is that almost 80% or 75% of tumors from ovary and colorectal cancer origin and breast, for instance, overexpress very much this enzyme. And it is known, and this was demonstrated. Here is the data. So there was no slide. I just moved it. Now I remember. So this is what I was saying now. Yeah? So it has been demonstrated that if you knock down this ALOX5 pathway in leukemic cells, cancer stem cells from leukemia, and they go down, and so they get killed. So you have a better response to treatment. It is also important that ALOX5 inhibition in any case seems to affect the normal hematopoietic stem cells homeostasis. So this is something that makes the inhibition of this pathway perhaps more attractive in the sense that in principle, it might have less adverse effects, adverse reactions or toxicity that if you use another one. No? So at this point, there's two ways that we know about how to inhibiting ALOX5. One is to use Xyloton. Xyloton is a drug that is being currently used for asthma treatment, so it's an anti-asthmatic. Or by using siRNA sequences against the ALOX5 gene. You know? So if you actually perform, well, this is one of these omic expression study and in cancer stem cells from different breast cancer models. And the only thing I wanted to show you is that this is one of the targets that often appears the most on top of the list as overexpressing cancer stem cells. And this affects these two types of different breast cancer cells, which are the MCF7 and the MD231, which is interesting in the sense this is ALOX5, for instance. What you see here is what happens if you compare the levels of ALOX5 in cancer stem cells and in tumor cells. So if you allow me, I will refer to cancer stem cells as the cancer stem cells of a given tumor and non-cancer stem cells as the rest of the cells. Make it a little bit easier. So this is interesting because this cell line is very different than this one in the sense that this is very mesenchymal, so more cancer stem cell-like, let's say it in that way, and highly resistant to treatment. This is more epithelial, has a much smaller percentage of cancer stem cells inside, and it is more sensitive to treatment in general terms. Okay, so but the same thing happens if you compare other cell lines like these two ones that are from colorectal origin. In any case, you always find much higher levels of ALOX5 in cancer stem cells than in non-cancer stem cells, which fits a little bit with what was found also in leukemia cells, for instance. No? So what happens if we use Xyloton, for instance, to treat these cells? And the most interesting thing here is that, first, cancer stem cells are more sensitive to the inhibition of ALOX5 with Xyloton. But the most interesting thing here is that when you have more efficacies in this case, which is when you grow cells as mammospheres. So if you take these cells and you grow them in non-attachment conditions, what you will see is that cancer stem cells are the one that survive and the one that makes these tumor spheres. So you can actually monitor how these mammospheres behave, and you can actually test the drug in that situation. No? So what this is telling you is that when you have mammospheres, because cells are growing in non-attachment, so mainly cancer stem cells, is when you will see the more efficacy of this inhibition. No? More even if you have cells in attachment conditions like in this case, or in this case here. No? So it means that as a proof of concept, it might work. So we have Xyloton, we can use it, we can inhibit ALOX5, and we can actually kill cancer stem cells. No? So what happens if we use an siRNA sequence? So the same thing. So this is survival of cells. If you knock down ALOX5 in cancer stem cells, also the survival of these, the percentage survival of these cells, they just go down dramatically. No? So this means that we can use at least these sequences or this compound just to try to do something. Let me tell you something. This is interesting in the sense that either if you use Xyloton or this ALOX5 siRNA, at the end, what's more important is that you don't see this. And what is this? This means that if you treat cells with paclitaxel, for instance, or Abrexan, for instance, which is, as you know, this albumin paclitaxel nanoconjugate, what you see, if you monitor the percentage of cancer stem cells in a cell line, for instance, what you see is that even though they have efficacy, the percentage of cancer stem cells increase. Why is that? Because they're more resistant. It means that one thing is that you might be capable of killing cells. The other thing is that non-cancer stem cells are more sensitive, so we'll kill much more non-cancer stem cells than cancer stem cells. So at the end, the percentage of cancer stem cells is bigger at the end. And this is probably what represents the situation in which you treat a tumor, it shrinks, but still some cancer stem cells survive. Okay? So this represents that model. If you use Ziloton, the percentage of surviving cells is much lower, and even more if you use the siRNA sequence, probably because the inhibition is higher. So this means that by inhibiting ALOX5, the sensitivity of cancer stem cells get increased. It's not that you just can kill it, that you can kill those cells, it's just that they also become more sensitive, okay? So the question is, how can we deliver this by using nanoconjugates, and why we should do that? No? So one main reason is that it is very difficult just to treat a tumor by injecting siRNA sequences in the bloodstream. It's not going to work. So we need to have a delivery system for that. But the other one is that Ziloton by itself, it has a very extremely high IC50. It means that we need to inject so much quantity, so much amount of Ziloton in the bloodstream that it's not feasible. So at this moment, for instance, Ziloton is being administered by oral administration or by inhalation only for asthma treatment. So even though it might be useful as anti-cancer treatment, it's not feasible. No? So that's why we thought about using a polymeric system just to build up a nanoconjugate. In this case, we're using these polymeric micelles, which are made with amphiphilic polymers. So these give us the chance to have this hydrophobic core compartment in which we can actually encapsulate the Ziloton because it's highly hydrophobic too. Right? So this is some data about the size and polydispersity and the time data on these polymeric micelles with Ziloton. The good news here is that after a month, and now we know that after almost 3 months, they remain stable. So at least in size and size distribution, they're quite small, as you can see here. So they are around 20 nanomolar. And with a very small, quite acceptable level of dispersion. So one good thing is also that we can control the entrapment of Ziloton. This is due to the fact that we can actually use ferric chloride just to make this kind of reaction. So at the end, we can quantitate the amount of drug that gets encapsulated, and I can tell you that it's around 97%. What happens if we use the polymeric Ziloton and we repeat the same thing that we were doing before? So what we see is that the efficacy of the polymeric Ziloton is much higher even than the free Ziloton in this case. So it means that the internalization of the Ziloton in this way probably it's making a difference. So at the end, we have more efficacy in this cell line, and the same thing happened with the MCF7 parental. So at the end, we're seeing here that Ziloton, if it's administered by the polymeric micelles, it has much more efficacy in cancer stem cells, and this is done in cancer stem cells. So on the other hand, the toxicity of the polymeric micelles, of these polymeric micelles without drug is very low. So that's good. What happens if we monitor the cell-independent growth of these cells? This is the most important thing, because this is what's giving you the idea whether you're knocking down actually, or you're killing actually the cancer stem cells. No? So you see the same thing. So the polymeric Ziloton in vitro, it works much better than the free one. No? So that's good. No? So what if we use another way to inhibit the ALOX5, which is using the siRNA sequence? Well, we have to modify a little bit the polymeric micelle because we have to use cationic polymers just to make these polyplexes with the siRNA. So, but we still can put all these polyplexes here in the aqueous, in the external part of the polymer, and try it also in vitro. This is the data, so more or less it's the same thing that we were seeing before. The encapsulation, the entrapment efficiency in this case is very high, it's almost 97%. Sizes are more or less the same. Dispersion, excuse me, here is also very low. So the most interesting thing of this case is that for some reason the internalization of these particles into cancer stem cells seems to be much better than in non-cancer stem cells. No? So for some reason that we don't know, to be honest, it seems that cancer stem cells are more eager to entrap and internalize these polymeric micelles. And this is good because it might be the reason why we have more drug or more siRNA sequence inside and more efficacy. So what happens if we test these in different concentrations in the polymeric micelles in vivo? So we're talking here about the maximum tolerated dose. So, and we test different concentrations, and up to now we did not find the maximum tolerated dose. So any one of these concentrations did not show any kind of body weight reduction or anything that you see here that might be considered as a preliminary toxicity in the animal. So this is an example of how to use these polymeric micelles, for instance, with an siRNA against GFP. This is green fluorescent protein. So it's a protein which is fluorescent and is green. These are cells that express this fluorescence. If you use the polymeric micelles with the siRNA, so at the end, it's very effective in inhibiting the siRNA sequence. This was a proof of concept that we might use this system just to knock down the specific gene. And therefore, we did the same thing, this time with ALOX5s, for instance, in tumor cells, checking invasion in these two cell lines too, and also the level of expression of the ALOX5. So as you see, in vitro, it works pretty much. It works very well too. There's a reduction of the expression of the gene. In some cases, like in this one, this reduction is even better. But at the end, we have a reduction of invasion of these cells, and at the level of cancer stem cells, we have also a very strong reduction in cell growth independent assay. No? So we did not follow further from this point because we were interested in seeing, well, if the siRNA is working, seems to work fine, and the xyloton is also working fine. So what happens if we combine these two things? So we design another polymeric micelle with everything. So in a way that we can deliver xyloton and at the same time the ALOX5. And what you see here is that the combination of xyloton and ALOX5 has a much stronger cytotoxicity than ALOX5, than xyloton or ALOX5 separately. So it means that most likely putting together these two strategies in the same polymeric micelle, it might work much better. If you question yourselves why don't administer these two separately, the reason is that the amount of drug that needs to be injected and in vivo, it's too high and it's not feasible. And I'm going to stop here. Sorry. This is the people that has been involved in this project. This is still ongoing. Now there are in vivo tests being ongoing in the laboratory. So Diana is the PhD student that has been working mainly in the chemical design of the compounds. Peter Janet has been working very much in what's all the models on cancer stem cells and testing the systems, and Iván Evasolo and Yolanda Fernandez are being very much involved in what's everything related to the toxicity testing in vivo and in vitro of this system. So thank you very much. If you have questions, I'll be more than glad to answer them. Right there. Thanks.
Thanks for the presentation. I wonder if you have more information about the structure of the micelles, the different blocks that you used, the aqueous solubility of the drug that you are using, and what's the encapsulation weight by weight drug polymer?
You mean the drug loading capacity?
Yes.
No, I don't have that data here. I don't recall in that case. So I cannot give you too much details on that sense, but I can, if you're interested, I can send it to you. Yeah, sure, for sure.
And the structure of the micelle, do you have?
The micelle, this micelle was made with Pluronic, with 127, so that was the officially polymer. It was very simple. But with Xyloton, since it's very hydrophobic, it was easy just to combine it and to encapsulate it, yeah.
Here.
Where? Okay.
Wonderful. So I have two basic questions. First of all, can you hear me?
Yeah, sure, sure. Go ahead.
First of all, the MCF7 you selected, and you selected the total MCF7 cells, but those are mostly ER dependent breast cancer cells. So when you're talking about the cancer stem cells from MCF7 cells, did you select those kind of population, or this is overall?
Yeah. So. So one of the things that we did, and I didn't tell you about it because I said it many times before, so is that we use a specific methodology that we developed to track down these cancer stem cells. So we design a genetic system that is helping us to track them down because these cells become fluorescent. So in a very selective way. So when a given tumor cell becomes cancer stem cell, or if you have cancer stem cells on those tumor cell lines, they are bioluminescent, and we can, well, they're fluorescent actually, because the expression of a tomato, of a fluorochrome, which is called tomato, which is red. It means that we can track down these cells in vitro and vivo, so we can, let's say, count how, and get these percentages of how these cells react. No? So, and in that sense, I can tell you that it's true that the MCF7 cell line is very different from the other one, but if you go to cancer stem cells, then these differences almost doesn't exist. They look very much the same. Yeah.
Exactly, that's the point, because when you are doing IC50, it's so high, like around 300 to 400 micromolar, Xyloton. It's very hard to convince the FDA to go for clinical practice.
Yeah, that's the problem of Xyloton, that you need so high the amount of I mean, the efficacy is so high that there's no way to use it in real practice. No?
So on the other hand, but if you selectively, you can sort it out the stem cells and show that it's specific for stem cells, maybe your data might change. I don't know, I'm just curious, because you are taking the whole cells, maybe it's not killing normal, cancer cells, but just specific for stem cells. And maybe 3% or 4% based on what kind of cells you're working on it. So then the whole IC50 will change if you isolate it first by sorting.
No, in this case, so the reason why we designed this methodology is because we don't want to take out the cancer stem cells from the total amount.
So the data, you showed all the isolated cancer stem cells?
Some of the data is with, they're not with, well, some of the data is with isolated cancer stem cells, but it's not that we isolate them, it's that if you take the cell line and you grow them in non-attachment conditions, you'll see the cancer stem cells survive and they form these mammospheres, okay? So they are the red ones, let's say, But we like to work with this method because we don't separate cancer stem cells from non-cancer stem cells at the end. So the interactions and the cross-talk interaction between cells is kept, So it's a more, let's say, not natural, because those cell lines are being used commercially for many years, but it's a better way just to see how we affect tumor cells and at the same time the cancer stem cells. So, but it's true that at the end what we would like to see is that we kill cancer stem cells. So we're not so worried about the other cells which are not cancer stem cells, because eventually they die.
Wonderful, thank you.
Okay, so let me change.